Today, I'm going to talk about DNA replication process from chapter 5. DNA replication process occur on double helix of DNA. Firstly, before start the replication, the double helix DNA need to unwind to be the replication bubble. This is the replication bubble. By using the first enzyme that we call helicase. Okay, helicase function is to unwind the DNA by breaking the hydrogen bond, while the hydrogen bond is between two complementary bases. That means it's between this strand and this strand. Helicase unwind in opposite direction. The others will unwind to this direction, while the others will unwind to the other direction. After unwinding the DNA, it will form the replication forks. Replication forks are there, here, and here. Next, when the DNA is formed as a single strand, they easily to recall back. So, to prevent that thing from happen, so we need to use SSBP. That is the single-stranded binding protein. This is the function to prevent the recoiling at DNA strands. SSBP or single-stranded binding protein will attach the single strand of the DNA to not form back to double helix, because now they're on replication bubble. After that, we proceed with third one. That is the topoisomerase. Topoisomerase function is to relieve strands at double-stranded DNA. When the helicase are going to unwind the DNA, in the end of the replication forks, they will need to form strand. Thus, to form this strand, we need to use topoisomerase. Okay. Before we going through the replication, everyone should focus and understand how the new strand replication occurs. The replication of new strand occurs at 5' to 3' direction, while the old strand and template strand is from 3' to 5'. So after we understand that, we proceed replication. Start by forming RNA primer. RNA primer is formed by primase. Primase function is to synthesize RNA primer at the 3' end at template strand. 3N template strand. Next, let's see the picture. So, this is a 3' of template strand, while this is the 5' of template strand. Now, I'm going to talk about RNA primer. RNA primer formed at 3' strand. RNA primer consists RNA nucleotide. For example, A, U, C, and G. After RNA primer form, DNA polymerase 3 have to add DNA nucleotide. Because the function of DNA polymerase 3 is to add new DNA nucleotide complementary with template strain. The elongation of the DNA nucleotide at the end of 3' because the 3' end have 3 hydroxyl group. Before we proceed to DNA polymerase 1, we should know that the elongation of this strand toward to replication fork. The strand that towards to replication forks we call leading strand. We have another strand, that is the lagging strand. So it's the same like the leading strand. First, like primary RNA primary have a RNA primary is AUCG, then have a DNA nucleotide is formed by DNA polymerase 3 and ending with 3' because the 3' and have a 3 hydroxyl group. Okay, this elongation strand is away from replication fork. So the elongation strand is called lagging strand. So now we can talk about DNA polymerase 1, that the function to replace the RNA nucleotide to become DNA nucleotide. So we see now that is the RNA nucleotide. So we look again at this picture. This now DNA, this is a RNA nucleotide. Is A, U, C, and G. So DNA polymerase 1 will change the RNA nucleotide to become DNA nucleotide, which change U to T. So now from AUCG to ATCG, so that's mean the RNA nucleotide become DNA nucleotide. So now that's all is DNA nucleotide. Okay, last but not least, we talk about ligase. Ligase function is to join Okazaki with capital O. O is a capital letter. Fragments. To join Okazaki fragments. Okay, so we back again to the picture. So we make another fragment, just like this. I'm already picturing that it start from the polymerase that form RNA primer, and then DNA polymerase 3 will add a new DNA nucleotide, and then the RNA nucleotide will become the DNA nucleotide by using DNA polymerase 1. So this fragment is called Okazaki fragment. So now we see is the same direction as in the same like this one. Okay, next two Okazaki fragment will join by DNA ligase by forming phosphodiester bond. So now we see that okay, now we see that this replication proof that it followed by semi-conservative theory, but because when a new strain form, it recall back with the template strain. As you can see that this strain is recall with this double helix DNA, and this strain is recall with this double helix. So you see now that is the proof that this all is followed by semi-conservative theory. Okay, so that's all for my video. Thank you.